So, Greg, we're coming to an era when it looks likely that we can choose a particular part in the genome and replace it with whatever we choose. What's that called? What's the technology?
Genome editing.
And CRISPR Cas9 is the jargon for the technique?
That's the most recent and seems to be the most effective and powerful way to do this.
So
yes.
CRISPR sounds like something you use in the kitchen.
Yeah, it's not something to do with bacon. CRISPR is an acronym, and it really is a long name for the bacteria's immune system. So these
bacteria have immune systems.
Well, not like ours, but they have pathogens too. So bacteria get infected by viruses.
Huh.
So the CRISPR Cas9, the CRISPR enzyme is part of a system that the bacteria use to purge their genomes of pieces of virus that have gotten stuck in there.
So how does it work? A virus comes into a bacterium, and then what?
The virus has a different sequence of its DNA, and sometimes the virus will insert itself into the bacteria's genome, and what the bacteria does is keep track of all those foreign sequences and make a little library of them. And to make that library, it has to edit its own genome. And so it puts those little sequences in a certain place, and this is the enzyme that helps it do that. Then if that virus reappears, it knows that it's if the sequence is in the library, that's not a good thing, and so it attacks that sequence.
So it literally chews it up?
Chews it up, degrades it.
And what's the Cas9 part about it?
The Cas9 is a piece of RNA that is complementary to the DNA that is going to be replaced.
Complementary, so it locks onto the right place.
It locks onto the right place. We call it a guide RNA because it tells the enzyme where to do the cutting.
So far we've got something that can identify a place and cut something out, but you have to put something else back in. You
have to put something back in again, and so there's an enzyme system for doing that. And then not only does it have to be inserted, then the ends have to be sealed up again, and there's another enzyme for that. But those are enzymes that cells have to repair natural damage to DNA anyway. So the really special...
It's taking advantage of the system that's already there to repair DNA.
Exactly.
And you feed it something that's not what it was expecting.
Correct, yep. Yep.
Fascinating. So I hear about these techniques being used possibly to even drive a certain species of mosquito extinct or something, but it seems to me they may have much, much larger possibilities.
Absolutely. I think the big promise for the CRISPR-Cas9 technology is in therapeutics and the ability to potentially correct a genetic disorder that someone is born with by perhaps removing stem cells, editing them, and replacing them in the body. And
so muscular dystrophy is a dramatic one,
yeah.
And I've been interested in that because there's a good evolutionary reason why there's a vulnerability. The gene is gigantic,
yes,
compared to most genes, and yes. So it gets more vulnerable.
More opportunity to be mutated, yep.
So with CRISPR-Cas9 technology, how does it work to find the bad spot in the gene?
So what we would have to do in a case like that is know in advance what the particular problem is.
And there might be lots of different spots in a large gene like that.
Indeed there are. And so we know that certain problems with the muscular dystrophy gene, the dystrophin gene, run in families. And certainly we can take a cheek swab from an individual who's suffering from muscular dystrophy and figure out what their particular
so it really has to be personalized replacement. It's not a generic kind of thing.
Absolutely. So this would be tailored to fix the problem that individual has. Yes.
Now in cystic fibrosis, a high percentage have the same abnormality. Is that right?
Correct. So muscular dystrophy is a little bit unusual, and it's for exactly the reason that you allude to. This is a big gene that it's a bigger target for mutation, we say, and so they're sporadic changes that might only exist in a single family. Mm-hmm. With cystic fibrosis, it's a much more common mutation, and that's probably because that mutation actually has a side effect that's slightly beneficial under certain circumstances.
What kind of benefit?
Well,
it has something to do with sodium transport.
Correct, and this may have been protective against some sorts of pathogens in the past. So.
Salmonella typhi, for instance.
Potentially, yes. It's actually
or cholera.
Probably one of those. Yes, something that because of this benefit, that mutation was spread through the human population. So more people have it. Yeah.
So I've been thinking about this a lot, and it finally got through my head after thinking about this. We keep thinking about alleles being good or bad,
right?
And being either spreading or going extinct. And it struck me that this might well be one where, yes, there's a lot of mutations that happen that are gradually weeded out. Yep. However, they're not weeded out quite as fast because it might be small effects in a few individuals occasionally that give small benefits. Does that make sense to you?
Yes, and that's probably really the way this happens. So you mentioned 2 or 3 factors there, small effects. If something only has a mild negative consequence, it won't be eliminated as quickly as opposed to something that's really devastating. You also alluded to conditionality, is what we would say, that the mutation might only really have a consequence, perhaps, let's say, if you're malnourished or you're infected with a certain pathogen, but otherwise, perfectly fine. So selection won't really eliminate it very quickly under those circumstances.
Good.